Hi team! Today we're going to be discussing neural communication. What I talk about today will partially overlap with what you read about, but mostly extend on the readings. I also want to point out why we study neuroscience as part of sensation and perception, right? Like, why don't we just look at illusions all day? Well, the electrochemical action that's happening in our brains is the starting place for all of our perceptual experiences. We cannot understand perception without it. However, I've designed this course to focus on and emphasize perceptual processes rather than neural ones. So we cover enough neuroscience so that we can understand perception, but not more. Okay, let's get started. Wait, what's that sound? Oh, hey little buddy, what's going on? You look kind of sad.
You know what? I am sad.
Oh, what are you feeling sad about, little guy?
I feel like nobody understands me.
Oh, that's a tough feeling. But you know what? I've got great news. I'm about to teach these students all about you. And so then they will understand you.
Really?
Yeah, so pull up a chair. You can listen.
Hooray.
Yeah, that's how I feel too. All right, so I want to start off with another quote today. Which of our brilliant early philosophers was this? So Morpheus makes a really good point. What we are experiencing as reality depends on these electrochemical changes happening in our brain. Because of course, the brain can't actually feel or taste or smell. All the brain can do is interpret the neural code that gets to it. So therefore, we need some mechanism for converting the aromatic molecules that are released by a rose, or the sound waves that come off a piano, or the chemicals in pizza, into a code that the brain can understand. So the reading gave you a sense of what neural code is, but not much of how it happens. That is the process by which pizza becomes action potentials. So when we study each sense individually, we'll talk about that process. Today's goal is talking about some of the basic properties of neural communication that all senses have in common. So in addition to the types of neurons that you read about, there are also sensory receptors, which are the specialized neurons that come in contact with the sensory stimuli. So the simplest example of this is in our sense of taste. Taste receptors cover the surface of your tongue. Some taste receptors are particularly sensitive to dissolved salts and have ion channels that allow sodium to enter them, which cause those taste receptor cells to depolarize. This depolarization leads to EPSPs, excitatory postsynaptic potentials, in the neurons that synapse with the taste receptor. So I'm going to teach the general principles of the nervous system first to have that foundation firmly in place for when we move on to transduction in each of our sensory systems. So I'll now do a really quick recap of the reading to give you an opportunity with more visuals and more examples and detail. Okay, so as you read, at rest, there's a higher concentration of sodium outside the cell than inside it. And there's a higher concentration of potassium inside than outside. Note that both sodium and potassium are positively charged ions. So the fact that the cell is negative at rest doesn't have to do with sodium and potassium. It primarily has to do with the presence of large negatively charged proteins, shown here in teal, that are hanging out inside the cell. Okay, so this is what happens when the cell is just hanging out, unstimulated, at rest. When a sufficiently strong stimulus happens, that peaceful rest begins to change. Stimulation can happen in many different ways. Let's stick with the example of taste for now. You eat something salty, and the sodium ions on your tongue enter taste receptors on the tongue and cause them to depolarize. So the cell gets a bit more positively charged. When it crosses a threshold, that is the point, the level of depolarization at which an action potential occurs, that causes voltage-gated sodium channels to open. Because there's a higher concentration of sodium outside the cell than inside it, those positively charged sodium ions enter the cell and they bring their positive charge in with them, causing the cell to become more positively charged. That depolarization causes voltage-gated potassium channels to open. Because potassium is at a higher concentration inside the cell than outside of it, those gates being open means that potassium now rushes out of the cell. Because potassium is positively charged, when it leaves the cell, it brings its positive charge with it, leading the cell to repolarize or become more negatively charged again. Then there's this brief period of hyperpolarization before the cell returns to its resting potential. But at this point, you might be concerned that although the charge is back to resting, the level of ions are all messed up relative to how it was before the action potential, right? We got sodium on the inside, we got potassium on the outside. Who ever heard of such a thing? So we get back to our pre-action potential concentrations of ions via the action of a process called the sodium-potassium pump. This moves sodium out of the cell and potassium back in. We have some actual footage of that process in action. This is what it looks like. All right, let's watch another example of the action potential occurring. So here we go. Sodium channels open, sodium brings its positive charge in, depolarizes, potassium brings its positive charge out, hyperpolarizes, and then returns to baseline. What we've talked about so far in action potentials is in one spot on the axon, right? What's happening in one particular area. But action potentials are an ongoing process that move down the length of the axon. So the positively charged ions at point A, sodium channels opens, sodium comes in, those positive ions are now in at point A. Those diffuse down the axon, and that is what causes the voltage-gated sodium channels at point B to open, meaning that now sodium is rushing in and further depolarizing the cell at point B. That process continues such that there's a traveling wave of depolarization that propagates down the length of the axon. Here's what it looks like. So the flashes of light here are the action potential. You can see it's traveling down the length of the axon. As the action potential passes, ions on the outside, the sodium ions are what's being shown here, come into the cell, right? As the depolarization happens, it's happening because the sodium is entering the cell, bringing its positive charge in with it. Then after the action potential passes, the sodium moves back to the outside of the cell. When the action potential reaches the terminal buttons, reaches the end of the axon, it causes neurotransmitters to be spilled out into the synapse, into the synaptic cleft. And those neurotransmitters bind to ion channels on the postsynaptic cell, which open and allow ions to enter the postsynaptic cell. So the neurotransmitters that are released by the presynaptic cell can result in EPSPs, excitatory postsynaptic potentials, or IPSPs, inhibitory postsynaptic potentials, in the postsynaptic cell. All right, so the first thing we get a handle on is how neurons carry a binary signal. That's the action potential. And that binary signal is happening within the cell. The next thing we need to understand is how the signals from multiple neurons are combined together. So in our schematics, we typically see one neuron synapsing on one other neuron. But in fact, a given neuron may have connections to over 1,000 other neurons. So the postsynaptic cell isn't just getting input from one neuron, it's getting input from a whole team of presynaptic neurons. So it needs some way of combining the signals that it gets from those presynaptic neurons. Now, in different places in this course, you'll read about two terms that are associated with the relationship between presynaptic cells and postsynaptic cells. The first of these is neural convergence. This refers to the physical arrangement of the neurons. So these presynaptic cells are converging on a single postsynaptic neuron. The other relevant term is neural summation. So this refers to the process by which the presynaptic cell combines signals together. So that's like the process of combining the EPSPs and the IPSPs together. So convergence is about like the physical arrangement of the cells, and summation is the process by which the signals from those cells are combined. Let's work through some examples. Okay, so we have 4 different presynaptic neurons here. The excitatory ones are shown in pink and the inhibitory one is shown in blue. And then the star of our show is the neuron in the middle. This is the postsynaptic cell. So we're gonna look at how activity in the presynaptic cells affects activity in that postsynaptic cell. So in the first scenario, our postsynaptic neuron gets one EPSP from one of the presynaptic cells. So we've got activity in one of those excitatory presynaptic cells. And as you can see, that activity is not sufficient to cross threshold and lead that postsynaptic cell to fire an action potential. But if we have simultaneous activity from multiple of those presynaptic cells, then that combined depolarization is sufficient for that postsynaptic cell to cross threshold and fire an action potential. So one of the ways that we can get an action potential in that postsynaptic cell is by getting input from multiple different presynaptic cells. Another way we can get an action potential in that postsynaptic cell is rapid fire activity in one of the presynaptic cells. So that may be that there's a particularly strong stimulus that is affecting that presynaptic cell, and that leads the postsynaptic cell to be sufficiently depolarized to trigger an action potential. Okay, but let's look what happens when there's activity in the presynaptic cell that has an inhibitory relationship with our target cell. In that case, the target cell is hyperpolarized. So not only does it not have an action potential, but it's even harder for other excitatory cells to trigger an action potential in it because the cell's charge is even more negative than it was to begin with. Okay, finally, here's a situation in which we have simultaneous EPSPs and IPSPs. So one of the neurons is leading our target neuron to become hyperpolarized. One of them is leading the cell to become somewhat depolarized. And together, the change in charge that they have is negligible. All right, so we convert physical energy into neural code, and we combine the responses of many different neurons together. Our next process that is common across all sensory systems has to do with how neurons respond to a consistent stimulus. Just because a stimulus is consistent doesn't mean that our perception of it is consistent, right? Imagine jumping into a cold pool. It feels really cold at first, but over time, you kind of get used to it, right? The water hasn't warmed up, but it feels warmer. That's adaptation. So in nearly every sensory system, neurons respond less robustly to a consistent stimulus. So here's a schematic showing the baseline firing rate of a particular cell. When you first experience a sensory stimulus, you show a strong neural response, right? Your cells are saying, wow, something has changed here, something is happening. But then over time, that neural response reduces. So this happens in most sensory systems and is responsible for many of the fun illusions that we'll experience this term. Okay, so let's see an example. I want you to stare right at the black dot in the middle. Don't move your head, don't move your eyes, try to blink as little as possible. As you stare and stare, you may notice that the colors seem to fade away. If you move your eyes around, boom, they're back. Okay, we'll talk in more detail about the specific process by which this occurs in the vision unit, but the basic principle is that the light-sensitive cells in your eye are adapting to a consistent stimulus. So why doesn't this work for all things? Like if you just stare at me on the computer screen, I don't disappear, right? It works particularly well for this image because first, it's stationary, and because all of the images, the edges are fuzzy. So when the edges are sharp, any slight eye movement changes where the image falls on your eye, and that registers as a change. But when the edges are fuzzy and you stare at a single point, those small motions aren't coded as being terribly different. Okay, so we need, there's one more thing that every sensory system has to do. They have to get the information from the sensory receptors, from the sensory organs, up to the brain. So once the physical stimulus has been translated into neural code, then that neural code has to make its way up to the brain. So these yellow lines here represent major nerves or bundles of axons that carry information to the brain. So if you touch something hot, those uh-oh pain signals travel via axons to the spinal cord and then up to the brain. The same is true for things we taste and smell and so forth. So these are like one-way superhighways carrying information from the periphery up to command central. In the brain are primary areas of sensory processing for each sensory system. After being relayed through the spinal cord and brainstem, signals are first sent to these areas of primary sensory cortex. That is, the action potentials leaving the optic nerve of the eye relay or synapse several times and then are conveyed to primary visual cortex at the back of the brain. From primary visual cortex, information then travels elsewhere. You may recall from Intro Psych that in a delightful evolutionary twist, information from the left side of the body is primarily coded in the right side of the brain and vice versa. So in the visual system, that means information in your right visual field ends up in left visual cortex and vice versa. In the somatosensory information, that means when you touch something with your right fingertips, that information is coded in left somatosensory cortex. There are a couple of exceptions. Smell, for instance, is primarily ipsilateral, meaning that information from your left nostril is coded in your left hemisphere. More on that later. All right, that's it for today. Press pause now, and I'll see you in class. All right, here's the answers for part 1. Go ahead and press pause so you can check those. This diagram is so bad. Okay, first, there's no such thing as a size of an action potential, right? Action potentials are a binary state. They are happening or they are not happening, so you can't ever have a big one. The way that we code for big or intense sensory stimuli has to do with the rate that action potentials happen, not the size they are. Second, this diagram should have a baseline firing rate. So even in the absence of any stimulation, there should be some neural activity. Third, for the stimulation, the stimulus that's shown in red here, that's happening below threshold, so there shouldn't be an increased firing rate relative to no stimulation whatsoever. And finally, the rate of action potentials should be faster for the green stimulus than the blue stimulus, given that the green one is more intense. So here's the correct diagram. Note that everything that is shown in the early black and orange and red is conveying the same baseline firing rate. Okay, so for number 3, our baseline firing rate is 2 in the arbitrary units that we've selected here. If we stimulate receptor 3, that's going to cause an increase in the firing rate of our target neuron because neuron 3 has an excitatory relationship with the target neuron. If we stimulate 3 through 5, that's going to lead to even a higher firing rate because 3, 4, and 5 all have an excitatory relationship. If we also stimulate number 2, that's going to lead to a slightly lower firing rate than 3 through 5 being stimulated because neuron 2 has an inhibitory relationship with it. And finally, if we stimulate 1 through 7, that's going to lead to a firing rate below baseline because both neurons 1 and 7 lead to IPSPs in our target neuron. Okay, if we only had, if we had no baseline firing rate, that means that the firing rate of a particular cell could only increase. There's only one direction for it to go. The fact that there's a baseline firing rate means a particular neuron can code for 2 different stimuli or can indicate 2 different stimuli depending on whether the firing rate increases or decreases. This actually turns out to be how we code for some components of color. So particular cells, if they're firing above baseline, indicate that red light is present. And if they're firing below baseline, it indicates the presence of green. More on that in the unit on color. So adaptation, the fact that we adapt, means that the perceptual system is prioritizing novel stimuli over sustained ones. If the same thing is happening in our environment over a consistent period of time, and it's not dangerous or harming us in some way, it probably means we can attend to that somewhat less so that when there's something new happens in the environment, we can give that our attention. Thanks for your attention, and I'll see you in class.